Hello everybody. So this is my first Lakes Committee meeting, and I've come to realize that a lot of you seem to have known each other for a long time as well. But this is my first meeting, and I thought I'd just introduce myself a bit. And I've been studying lake trout for the past couple of years with Chuck Kruger and Andrew Muir. I'm based at Dalhousie University, and my main supervisor is Paul Benson. And before that, my background's in population genetics and disease ecology. But I've studied vertebrates exclusively, but anything from birds, and spent 4 years in New Zealand studying avian malaria transmission and bats and white nose syndrome here and in Nova Scotia. And we've just got some funding to study the evolution of bird song using population genetics models. So I definitely bring a population genetics perspective to you, and I hope you find this talk is interesting. I'm very friendly, and I'd like to talk about trout later on today, because I need to gather as much information as possible from you in light of these recent findings. So the presentation I'm talking about today is based on an article that we just submitted to the Journal of Great Lakes Research. It's the special issue of the Northern Lakes. And my co-authors, of course, are Andrew Muir, Chuck Kruger, Kim Scribner from Michigan State University, and Paul Benson. And we were talking about this loss of genetic diversity and a possible collapse of genetic distance between the lake trout ectomorphs, or different forms of the lake trout. Okay, so the main purpose of the study is to investigate, of course, this the genetic diversity of the lake trout. And this is part of a long-term monitoring project, and our current study extends upon previous work by Guinan et al., and who had showed a loss of genetic diversity up until 1999. So the last data set on the genetics that was published was 1999, and we're carrying forward from that. The second part of the study is to investigate this potential hypothesized genetic homogenization of the ectomorphs, or what could be thought of as reverse speciation. And this carries on from work by Andrew Muir et al. On the geometric morphometrics that points to a possible ecomorphological homogenization of the morphs. So, a little bit of background on how they came to be. I mean, it's thought that the lake trout colonized lakes across North America from glacial refugia after the recession of the Pleistocene glaciers. The conduits were the proglacial lakes, and work by Wilson and Hebert in the 1990s using mitochondrial DNA showed that the lake trout probably originated from 4 main glacial refugia: the Mississippian lineage coming up from the southeast, the Atlantic from the east, the Beringian from the northwest, and then the Nahanian from a smaller isolated pocket in the west. Our current contemporary data using the same mitochondrial DNA primers shows that when you look at each lake, the different ectomorphs in each lake actually have the same mitochondrial signature. So to us, this is evidence that the morphs have actually diverged in symmetry. The morphs aren't from different origins, which means that since the recession of the glaciers, they've actually been diverging in symmetry through this process of adaptive divergence. So, but more recently, it's possible that they are collapsing morphologically. There's anecdotal evidence out there that the morphs are starting to look more similar. They're not as diverse morphologically as they were a few decades ago. And then a recent paper by Muir et al. Shows that, although you can statistically tease the morphs apart using geometric morphometrics and Bayesian statistics, as you can see in this principal component analysis, they actually do cluster into a highly variable single cluster. And this is a pattern that's being shown in other species around the world as well. I'll give you an example in a bit. So our research hypotheses are that lake trout ectomorphs have actually experienced a loss in genetic diversity, and that the genetic distance between the ectomorphs has collapsed through time. And in order to address those hypotheses, we were looking to, examine this loss of genetic diversity, especially since the 1990s, using microsatellite loci and historical data sets. And we want to determine whether the genetic distance between the ectomorphs has actually collapsed since the 50s. Where we have the historical data sets as well. And importantly, it was important to look at effective population size and see if that changes proportionally with these increases we're seeing in actual population size. So the data used in the study encompasses 3 time periods. The 1950s is our historical period, and the recovery period in the 1990s. That was contributed by Kim Scribner's lab. And this is work that's already been published, and we're adding the contemporary data sets from the 2000s. And we're looking at Isle Royal, Standard Rock, and Klondike and Caribou Reef areas, as you can see in the small location map from left to right in the red rings. So across the lake, it's essentially. And the results on the genetic data do confirm the hypothesis that there has been a loss of genetic diversity through time and since 1999. So even up through the recovery period. So what you're looking at in this graph is allelic richness on the y-axis is our metric of genetic diversity, and the x-axis is years, and we're looking at the changes in allelic richness through time. From before the collapse and then through the fishery collapse time period is going to be shaded in gray for a couple of other slides as well. Through the fishery closure and this incursion of sea lamprey, and then through the recovery period until now. And the first thing I want to point out is that historically, this first loss of genetic diversity period from 1948 to 1959, represents a 20% loss in allelic diversity. Through from the 50s to 1999, it's a further 10% loss. And then surprisingly, from the late 1990s, there's another 20% loss. These losses are all morphs and all locations to varying degrees. So, and the latter half represents a loss of about 4 to 6% allelic diversity per generation of lake trout, based on 7-year generation time. And overall, it's a 30% loss since 1948, which is quite substantial. Despite these losses in genetic diversity, though, there have been substantial increases in the actual population numbers. So this graph shows, it's a measure of relative abundance of the leans only we're talking about. Through time, it's data contributed by Sean Sitar, thankfully, from Isle Royale and Standard Rock. And you can see the Isle Royale leans, the numbers become extremely depressed during the collapse, but then they exponentially rise until the 1990s, where you see this plateau effect, which most likely represents they've reached a carrying capacity, and that's higher at Isle Royale than it is at Standard Rock, where there's a higher degree of fishing pressure. But importantly, from the genetic side, the effective genetic population size, which is the number of breeders, actually does proportionally follow what's happening with the actual population size. And that's good news, because you wouldn't want the number of breeders to be declining while your population size is growing. So they are breeding, which is a good sign. So the results I'm showing you here are the results from a Bayesian approximation analysis in a program called MSVAR, if any of you are familiar. But we're looking at the joint posterior distributions here, is what the scattered dots are, of the historical population size on the x-axis to the contemporary population size on the y-axis. And if that distribution is above the diagonal line and then to the left, like here in the leans, then that means there's been a huge change in population size. And as you can see, in the 1950s, because we're comparing 1959 to 2007, On a relative scale here, though, we've got lean effective breeding population sizes were quite low in the 50s, and they have increased substantially. For humpers and Siskiwet, the changes haven't been as great. Humpers might have increased a little bit, and it appears the Siskiwet have decreased a little over this period, but we still have to investigate that further. There could be something wonky with the data, and also, importantly, since 1999, though, there hasn't been a lot of change in effective population size. So you can see here, regardless of the location or the morph, they are all lying on that diagonal line, which I think is also a good sign of when an animal is in its recovery phase. Okay, so we've shown you that our data support a loss of genetic diversity, but the population sizes and effective sizes have rebounded. But now we need to see whether or not the morphs are collapsing. And the metric is genetic distance in a multiple co-inertia analysis. We're using the Euclidean distances, basically, between the centroids of the distributions for each morph. So you can see here, the leans are almost twice as distant from Siskiwet as red fins are from Siskiwet. So that's how those graphs are read. And here are the results. And these results confirm the hypothesis that there has been a collapse in the genetic distance between the morphs over time, and that it has intensified through time. So first, we'll look at the graph on the left, and it shows data from the 1950s for lean Siskiwet and humpers, and we're looking at the distances between those morphs. And you can see the distances are much, much smaller in 2007. And this represents about a 60% reduction in the genetic distance between the morphs. And then if you break it down on the right here, in smaller time periods, so from 1959 to 1999, the distances, or the differential between the distances, is actually quite a bit smaller. And the difference, this differential gets larger from 1999 to 2007, which represents that the rate of collapse has actually intensified through time. And I'd really like to talk to people about that because it does seem like we do have this scenario of reverse speciation is a bit of a coin phrase, but what all we're talking about is that you have these young species in post-glacial environments that are actually diversifying through the process of divergent adaptation, just like the lake trout. And then, as has been shown in many studies, also in many different places in taxa, human disturbance can have a homogenizing effect, whether you're homogenizing the prey or the water is undergoing eutrophication, and anything that can alter the selective pressures and basically reduce the effects that are isolating the animals. If they aren't isolated anymore, then you run the risk of intergressive hybridization, which possibly could have happened in this situation. And this causes a loss of genetic diversity and in turn reverse speciation. And I'm just going to show you one part of this graph here on the left in the study or the review by Seehausen and et al. And we're looking at the Enos Lake sticklebacks. You've got two hugely different morphologies, and in 1977 they were very different and they clustered separately in multivariate space. And then by 2002, not long after that, you see one highly variable cluster, just like I showed you in the beginning of the talk, the lake trout. This is possibly what could be happening with the lake trout, and the genetic data backs it up as well. So in conclusion, despite a market recovery in the population size, lake trout, depending on your morphin location, have lost about 20 to 36% of their genetic diversity since 1949. And the ectomorphs have collapsed in genetic distance over those three locations that I showed you today by 57%. And this has intensified through the years, and we can talk about it later or in the question period, but to me, it does seem very convincing that there is gene flow happening between the morphs, and they're introgressing to some degree. So it's not just a plastic change that's happening. So, and this might be, an indirect consequence of those population sizes being reduced and the loss of genetic diversity from the fishery and sea lamprey. But something else has happened since then. Maybe it's about community changes, something to do with the habitat, and maybe, I'm putting this forward, but maybe something to do with spawning behavior. I wonder if there has been a change. So our possible management implications that I want to suggest are that it would be good to separate management by the morphs themselves, because there could be different things going on, but especially the locations. And we've seen some other data today, especially the telemetry data, and genetically they don't seem to actually move huge distances. So our world is going to be completely different from standard rock. For example, protection during the spawning season might be important, and now there's more information about where the ciskowets are spawning, or that they spawn in early summer, and that might be something to protect. Again, have scenarios of gene flow drawn out if you want to talk about that later too, but it might be a good idea to protect that area, especially as the lean population rise. And it's always a good idea to be wary of stalking and the genetic consequences there. And we definitely want to prevent further loss of genetic variation by continuing to genetically monitor the breeding population. And of course, regulate the fishery harvest by ectomorph, and continuation of control of sea lamprey will be important in the future. And just from a research point of view, I think to me it would be really important to actually look at losses of genetic diversity at adaptive sites, and not just the microsatellites, but actually sites of under selection, because more likely than not they're probably losing diversity faster, and that's why you see a rapid collapse of morphology. But also the transcriptomic studies on hybrids might be important. Hybrids might not be so bad, but it would be good to understand how they are acting relative to pure weeds and pure ciskowet under different circumstances. So with that, is there any questions? Thank you.